Hello everybody, my name is Lee and you are watching B-Tech 8003 Communication Project. In 1997, the movie Gattaca described a not too distant future where social status is decided on whether a person is lab created, genetically modified or not. That seemingly sci-fi scenario has taken one step closer into reality in 2018 when He Young Kuit announced the birth of the world's first genetically modified babies, which provoked a world outcry and once again raised the question: human genome editing, is the world ready? Well, human gene editing has a pretty long story, but only really taken off in 2013 when CRISPR was first used in eukaryotic and human cells. CRISPR is a potent molecular tool that can cut, delete, and edit DNA with superior precision, usability, and cost compared to previous tools. It is a scientific breakthrough that rapidly transforms genetic study and generates a tsunami of research on genome editing. Human genome editing can be categorized into somatic gene editing, which is not heritable, and germline gene editing, which is heritable. Well, the hype on human genome editing is not unfounded, considering its potential in medical and healthcare, in which it can be used to kill sickle cell, cystic fibrosis, together with 10,000 other monogenic diseases, or to kill every infectious disease by simply delete the genome beyond repair. In fact, the first CRISPR-based therapy trial has been conducted directly on humans. Human genome editing has also potential in trait enhancement, where people can decide on the characteristics of their child up to their preference, creating a true designer baby. But this application is still highly controversial. Of course, the rapid progress in human genome editing has given rise to some concerns. The first concern is safety, since somatic gene editing can cause off-target edits, unwanted on-target edits, or unpredicted effects on the gene network. Germline gene editing can cause even more severe effects, such as mosaic and unpredictable effects on offspring and gene pool. After all, everyone has a different genome. The next concern is the accessibility of the technology. In case of limited access, this technology would only be available for a certain class of people, which might deepen inequality and facilitate genetic discrimination. In contrast, unlimited access would make everyone become the same genetically ideal, which leads to the lack of individuality and the sense of unauthentic and fake self. After all, even your personality might be artificial. But the biggest problem right now is how to draw a distinction and who to decide between good and bad uses, between disorders and traits, and between therapy or enhancement. Of course, it could be easy with HIV, sickle cell, or cancer, but what about dwarfism, deafness, or albinism? A physician scientist whose father of an albino, legally blind daughter said that doing so might have erased some of the things that make her special. Considering the risk, regulation has been put on human genome editing. There are even two international summits on human gene editing to discuss the problem. Somatic cell editing is permitted, but is put under strict regulatory framework. On the other hand, germline gene editing remained highly controversial and deemed illegal in many countries. Even He Yang Kui was jailed for 3 years for his conduct. However, the use of germline gene editing could be open to discussion once the civilization advanced enough. So, return to the question, is the world ready? Well, the answer is not yet. Not until we have better science knowledge, regulation framework, and public awareness of the matter. However, we can surely hope for a not too distant future where people can stay strong, stay healthy, and stay humanity. I would conclude the presentation with a line from the movie Gattaca. There is no gene for the human spirit. Thank you for paying attention.